everyone, thank you for joining this talk. My name's Sarah Steele and I'm a PhD candidate at Macquarie University in Sydney, Australia. And today I'll be talking about my research titled Modulating Auditory Feedback Loops Does Transcranial Magnetic Stimulation, or TMS, Induce Changes in Efferent Function? We know a lot about how sound travels from the ear to the brain, but much less about how the brain sends signals back to the ear. This feedback loop, known as the auditory efferent pathway, helps us focus on important sounds, protect our ears from loud noises, and adapt to complex listening situations. Understanding this pathway could have wide-reaching implications, as disruptions in this system have been linked to conditions such as tinnitus, auditory processing disorders, and sound hypersensitivity, all of which affect millions. And while there are ways to manage these conditions, there's currently no known cure. To move towards real solutions, we need better insight into the auditory efferent system. But that raises an important question. How can we study this pathway in the human brain? Well, we can alter brain activity in the auditory cortex and observe what happens in the ear. And there are a few ways to do this. In animal studies, researchers use cortical cooling, chilling the brain with ethanol and dry ice to disrupt neural activity and observe the effects downstream. But this method is invasive and not an option for human research, or for a PhD student like myself without neurosurgical training. In humans, researchers have used attentional tasks, asking participants to focus on sounds and measuring changes in the ear. Some studies show promising results, but the outcomes are inconsistent, likely because tasks and attention levels vary and are hard to quantify. We took a different approach and used transcranial magnetic stimulation, or TMS, here on out. TMS is a non-invasive method that uses magnetic fields to alter brain activity. So TMS involves placing a magnetic coil against the scalp, and that delivers pulses that temporarily change the activity of the neurons underneath. It's FDA approved, safe, and already used to treat conditions like depression, Parkinson's disease, and OCD. TMS is even used clinically to treat tinnitus. However, unlike in the motor cortex, where a visible response, which is a finger twitch, confirms effective stimulation, there is no immediate observable response that occurs when TMS is applied to the auditory cortex. Because of this, tinnitus treatment outcomes rely primarily on questionnaires to determine if the treatment is helping, since we currently lack an objective measure to verify whether TMS is affecting the auditory system. And that's where my study comes in. We tested whether TMS applied to the auditory cortex affects brain activity and, crucially, whether it causes measurable changes in the ear. We recruited 12 young adults with normal hearing and used EEG to measure brain activity and otoacoustic emissions, or OAEs, to measure activity in the cochlea. First, we established each participant's resting motor threshold. We applied TMS to the motor cortex until we found the minimum stimulation level that made their finger twitch 3 out of 5 times. This told us how sensitive each person was to TMS and helped us set safe, effective levels for stimulation in the auditory cortex. It was essentially how we calibrated the machine for each individual. Next, we recorded baseline activity. EEG electrodes were placed on the scalp and OAE probes were inserted in the ears. We got the baseline responses of the brain and ear activity before any TMS. We then ran our control condition, sham TMS. Sham TMS involves a coil that creates the same acoustic sound that a normal TMS coil will produce, but doesn't stimulate the brain because it doesn't create that electromagnetic field. Again, we recorded EEG and OAEs here. And finally, we delivered 40 seconds of active TMS to each auditory cortex at 80% of that individual's previously determined motor threshold. This was a repetitive TMS protocol where we delivered short bursts of high-frequency stimulation, and we followed this with another round of EEG and OAE recordings. During all the recording sessions, participants completed a click train task. Basically, they heard streams of clicks with an occasional louder deviant click that was 8 dB louder than the rest, and their job was to count the number of deviant clicks. So let's move on to the results. Participants correctly identified over 80% of the deviant clicks, confirming they were actively engaged in the task across all conditions. Now onto the EEG results. So we looked at the N1P2 response, a signal linked to how the brain processes sound. Surprisingly, there were no significant differences in brain responses across baseline, sham, and post-TMS conditions. even when looking specifically at responses to the louder deviant clicks, there were still no significant changes across the conditions. Here, each line shows one participant's response, and you can connect them across the 3 conditions: baseline in pink, post-sham in blue, and post-TMS in grey. And again, while we expected TMS to reduce that N1P2 amplitude, we didn't observe that effect. We also checked P2 latency, the timing of the brain response, and again found no significant differences there. But here's where it gets interesting, the OAE results. We did find a significant difference after TMS, but not after sham. So that showed us that the ear's response changed following real TMS stimulation of the brain. And our control showed that there was no response when stimulation did not occur. Looking closer here, we can see a clear difference. I've got a black arrow pointing to the difference between baseline and sham condition, and that sort of magenta arrow comparing baseline to post-TMS. And you can see that there is a significant change there. After TMS, there's a larger statistically significant difference compared to the sham condition. And here's another way of visualizing that. On average, OAEs dropped about 1.4 decibels after TMS compared to a reduction of only 0.7 decibels after sham. And this again shows us that TMS had a measurable objective impact on cochlear function. So why didn't we see EEG changes? Well, our setup was only a single channel array EEG setup, which may not have been sensitive enough to detect the changes that we saw. Importantly, though, we did see that even though EEG results were inconclusive, there were clear, measurable changes in the ear's function after TMS. This is really exciting because it provides the first objective evidence that TMS can influence auditory function, and this could open the door for its use in treating tinnitus and other auditory conditions. So our study lays the groundwork for future research. We now know that the brain can influence the ear via TMS and that we can measure it. And as part of my PhD, we're going to continue refining the stimulation protocol. We're going to use a more sensitive EEG setup with a 64-channel array, and we'll explore the optimal duration and intensity needed for TMS to target the auditory cortex. Thank you so much for listening, and if you'd like to discuss this work or anything else, please reach out. Thank you again.